Welcome. In this session, I'm going to begin with a new chapter, Biotechnology and its applications. I hope you have understood the principles and processes of biotechnology, right? So, where we are able to cut the gene of interest from the source organism by using enzymes like restriction endonucleases, and then able to ligate with the host DNA by using ligases. And so okay, so for which we need to use vectors. So there are various kinds of vectors, so which we have seen commonly the plasmids, so which were able to carry the gene of interest from the source to the host organism. Now in this chapter we will mainly focus on the application part of biotechnology. So let's see how I can define the biotechnology applications, right? So I say here there is a industrial scale production. So here it deals with the industrial scale production of biopharmaceuticals, biopharmaceuticals and biologicals by using genetically modified organisms. And the industrial scale production, which means producing in large quantities, not in small quantities, because we need to benefit the whole humankind. So for that reason we need to produce in large quantities, not in small quantities. So we need to produce, okay, whatever. So I told you that once we transfer the insulin gene into the bacterium and make the bacterium to produce insulin, I need to produce that in large quantities. Okay, so that's an industrial scale production of those biopharmaceuticals, biologicals by using the GMOs. I hope you we know that the GMOs is a short form of genetically modified organisms. So you understood what these genetically modified organisms, which means organisms whose DNA has been altered. So GMOs are organisms, okay, whose DNA is altered. So how we have altered? So we have introduced the foreign DNA into the host DNA, and that has altered the host DNA molecule. So they are all called the genetically modified organisms. So let's see how this biotechnology helps us. So applications of biotechnology. Some examples we will see before we discuss in detail, like in therapeutics. Okay, so in case of the therapeutics, we are using this biotechnology. So what we mean by this therapeutics? So these include various treatments, okay, for patients. So various treatments for, okay, patients is what we say the therapeutics here. So biotechnology will help in therapeutics. Then even it will help in diagnostics. Will help in diagnostics. What we mean by diagnostics? So these diagnostics will be including techniques to identify diseases. So techniques to identify diseases. See, not only just identifying the diseases, in our coming classes we'll also come to know that even before we get disease, is there any chance that we can come to know that I'm going to get a particular disease? So that is also possible with biotechnology. So a person can predict even before he gets a disease, right, with the help of biotechnology that I'm going to discuss in the coming sessions. Right, once we get the disease, to find out what kind of disease a person is having, that is what we call diagnosing. So finding out what disease the person is having. So in that also the biotechnology helps to find out with what disease the person is suffering from. Then developing GMOs for agriculture. So developing GMOs for agriculture. So we are able to produce various types of crops whose DNA is altered. So genetically modified crops. So we have developed genetically modified crops, so in which we can see that some of crops are able to live in drought conditions or with very less water availability, or can withstand high temperatures or pHs and whatever. So for that reasons we have basing on our requirement, so we develop these genetically modified organism or crops for agriculture. Then even we are using this in food processing. So using in food processing, so here food processing is that procedure where the transformation of raw ingredients into the food. So here making food from raw ingredients, like the manufacturing of potato chips, right, and many other kinds of foods that are already readily available in the market. Okay, so those are all will come under, some of them will come under these food processing processes. So making food from the raw ingredients, the food processing. And then we use even something called bioremediation. In bioremediation processes also we use biotechnology. So what we mean by this bioremediation, so it is simply the use of microbes to remove pollutants, So bioremediation is use of microbes to remove pollutants. And even we are using biotechnology in wastewater treatment. Okay, treating the waste waters before we release into freshwater bodies. Okay, and then even we are using in energy production. So these are, okay, some of the applications of biotechnology. Now in biotechnology there are three critical research areas. So let's look at those three critical research areas. So three critical research areas of biotechnology. So three critical research areas of biotechnology. So what are they? So one is providing a best catalyst in the form of improved organism. So providing a best catalyst, right? Okay, in the form of improved, right? So in the form of improved organism, and that improved organism usually it will be a microorganism or a pure enzyme. So either it is micro or a pure enzyme. So that will be the best catalyst. Okay, so an improved best catalyst, so that we get a very good result. Then we need to find out the best optimal conditions. Okay, so creating optimal conditions creating optimal conditions, okay, uh, you know through the genetic engineering, through the genetic engineering for the catalyst to act. So once we have prepared or improved, microorganism or enzyme, so it requires some, what I say, conditions, like what is the temperature at which it can grow very well, or what is the temperature at which this enzyme can work its maximum or the best, right? So different organisms will have different temperatures, enzymes also will have different temperatures at which they are best, okay. Now, to find out what a microbe or a pure enzyme works at what temperatures and all, so through that genetic engineering we can come to know, so creating the optimal conditions through the genetic engineering for the catalyst to work. So that is another research area where scientists are trying to find out all these requirements for that microbe. So at what conditions it can be grown very well, so that we can get the maximum product from it, right? Then, and third critical research area is the downstream processing, right? So this is another critical research area. So downstreaming processing technologies to purify a protein. So what are all that new techniques, methods that we can collect the product once we have created those genetically modified organisms? I told you in the last session that once we have introduced that insulin gene into the bacterium and made it to express, and that insulin will be released into the okay, nearby medium, that is into the nutrient medium, and we need to develop methods to collect that insulin from the nutrient medium, purify it, okay, and then so that we can sell it in the market. So all that techniques, so we need to develop new techniques in biotechnology. So in this step, that is downstream processing, so to purify a protein or an organic compound and so on. So these are the three critical research areas. So scientists are trying to develop, okay, best microbes or pure enzymes. So that is one research area. And another research area is that at what conditions those developed microbes or enzymes will act to their best. And then the downstreaming processes, so the downstream processing, where once we get the product, how we need to separate that product and purify that product and use it in, okay, for marketing purposes. So these are three critical research areas of biotechnology. Now we will see the applications of biotechnology in various fields. So almost in many fields we see the biotechnology is being used. So we'll see that uh, applications of biotechnology, in agriculture, applications of biotechnology in agriculture, then applications of biotechnology in medicine. Now let's see the applications of biotechnology. Okay, I will be discussing mainly in two fields. One, it is the biotechnological applications in agriculture. So biotechnological applications in agriculture. After that, I will be discussing the biotechnological applications in medicine. So how we are using biotechnology in agriculture and also in medicine. So first we'll discuss the applications in agriculture, that is biotechnological applications in agriculture. Now we have three options to get a better yield in case of agriculture. So what are those three options we are using? So one, it is agrochemical based agriculture. So in this agrochemical based agriculture, we will be using pesticides and fertilizers. So fertilizers and these pesticides are chemicals, and these pesticides are used to kill pests. So in this, the pesticides are chemicals that kill pests. And fertilizers, so these are synthetic fertilizers that increase soil fertility. So what happens is, as we continuously grow the crops in the agricultural field, there is a chance that most of these minerals, will be exhausted. So it means, what I say, the fertility of the soil gets decreased. So to bring it back, we use these synthetic fertilizers, right? And to kill the pests, so most of them are insects, to kill them, so we use these pesticides, right? The problem using these fertilizers and pesticides is that both of them cause environmental pollution. So of course, using them has definitely resulted in increase in the production. And another method is organic agriculture. Organic agriculture. So in this organic agriculture, right, the farmers like to use like these bio fertilizers and something called biocontrol agents. So here, instead of these biopesticides, the biocontrol agents, and instead of these synthetic fertilizers, we use these biofertilizers, which actually do not cause any environmental pollution. Okay, then the third one is GMO based agriculture. So it means that in this method of agriculture, we will be using genetically modified organisms. So what I'm going to do, by introducing certain genes into the plants, I can make a particular plant that is capable of self defense, right? Say suppose there are certain kinds of insects that can damage root, stems or leaves, right? And I can use certain kinds of genes from other group of organisms and introduce into these crop plants and make that crop plant, which is capable of killing that pest. So when these pests, that is insects, when they try to eat the leaf or stem or root, they will die. So how we are going to do it, I'm going to discuss those shortly in detail. Okay, so these are the three options. One is agrochemical based agriculture, so that we are using this from very long back, right? And organic agriculture, so because we came to know that these two are causing environmental pollution. So these pesticides and synthetic fertilizers, they cause environmental pollution. So here in this organic agriculture, obviously there is no environmental pollution, and even in case of GMOs also there is no environmental pollution. And moreover, you need not even use like these biofertilizers or biocontrol agents. So in fact, the GMOs, I can make a plant that can live in a condition where the water availability is very less or in high temperatures, okay, whatnot. So there are multiple options that we have, okay, in this GMO based agriculture, right? Now let's see about this GMO based agriculture. As I told that I can develop the crops which are capable of living in drought conditions or in cold conditions and whatnot, depending on the requirement. Okay. Now, in what way the genetically modified organisms, okay, are useful in agriculture? Let's see the genetically modified. I simply say crops. So they are called GM crops, genetically modified crops. So here, any organism, before that we see what actually is this genetically modified organism. So any organism whose genes have been altered. So any organism whose genes have been altered. So they are all called genetically modified organisms. And what we have done, so we have, okay, modified the genes of bacteria. So we have the genetically modified organisms under bacteria. We also have altered the some species, not all, some species genes we have altered in case of fungi, also in case of plants and also animals. So their, okay, genes have been altered, okay, by introducing some other genes into it or by removing it, whatever. So we have altered them, and that altered organisms means their genes, whatever those organisms whose genes have been altered, so they are all called genetically modified organisms. Okay. Now let's look at the significance of GMOs. So what is the use of these GM crops? Significance of GM crops. Right. One, so they will be more tolerant to abiotic stresses. So I can develop crops that are able to resist, abiotic stresses. So that's what I say. The GM crops, we can make GM crops, okay, that are tolerant to abiotic stresses. So we can develop genetically modified crops that are tolerant to abiotic stresses. So these abiotic stresses like okay, cold, right, and the drought conditions where the water availability will be very less, and in very cool temperatures also we can make certain kind of crops to grow, right? And in salty soils, they are called saline soils. We know that the plants will not be able to grow if the salt is more in the soils, because there will be exoosmosis, and that results in the death of the plant. Okay, and even in heat, that is hot conditions, right? So that is what the genetically modified crops, we can construct them to withstand these abiotic stresses. Then we can also construct the genetically modified crops that have reduced dependence, that have reduced dependence on chemical pesticides, on chemical pesticides. So just now I have told you that in agrochemical based agriculture, we need to use these pesticides to control the pest. I can construct genetically modified crops that are capable of fighting with this pest by themselves. How we are going to do shortly, I'm going to explain that. So GM crops, we can develop GM crops that have reduced dependence on chemical pesticides. So those crops are called pest resistant crops. So I can say that we can develop using the biotechnology pest resistant crops. Then we can even develop the crops that have decreased post harvest losses. So GM crops that have decreased post harvest losses. So what we mean by this post harvest losses is that once we harvest, say suppose you have grown tomatoes in your agriculture, and right, once you have harvested means you have plucked them from all the plants and you need to store them till you will be selling in the market. Now, by the time you sell, if anything delay in the days, then it can result in the spoiling of these tomatoes, right? But with this biotechnology, we can develop the, what I say, those in this case tomatoes, what I have said, with they will not get spoiled for months. So we can develop in that way, right? So that's what, if they don't get spoiled, then definitely there is a decrease in the post harvest loss. So that's what the GM crops that have decreased post harvest losses, so I can also do by using biotechnology. Then I can also develop GM crops that have increased efficiency of mineral usage. GM crops that have increased efficiency of mineral usage. Normally, when you are growing plants in the field, the plant will take up whatever that minerals present. Okay, whether they are useful or not useful, that doesn't matter. Only when there is a concentration gradient difference exists between the soil and the cells of the plant. So what happens is those minerals from higher concentration, they simply move into the lower concentrations, and the plant takes up the minerals even though they are not of use. So this will remove the minerals from the soil, and in a short period of time it can result in decreasing the soil fertility. So I can develop crops, that is genetically modified crops, that they will take up the nutrients only when they are required. So that is what the meaning. The GM crops, we can develop the GM crops that have increased efficiency of mineral usage. Next, I can also develop GM crops that will have enhanced nutritional value. So GM crops that have enhanced nutritional value. So I can develop crops, okay, where we see the fruits or whatever the food we get from those crops have a high nutritional value. Like for example, uh, vitamin A enriched rice. So vitamin A enriched rice also we can develop. Already scientists have tried it. They tried to make rice that have vitamin A, and they called it golden rice. But due to some limitations and whatever, it was not yet released into the market, and the scientists are still working out. Maybe if not in future, we will have this kind of rice in the market with the help of biotechnology. So we can develop the crops with enriched nutritional values. Already If not in this case of rice, already we have developed in case of some vegetables that I'm going to discuss it later, right? Already we have developed them with nutrition, with some vitamin C, vitamin A enriched, okay, the vegetables, and they are already in the market. So except this vitamin A enriched rice. Okay, so we can develop crops, so which I call them the genetically modified by altering the genes of that plants, right, and can make enhanced nutritional value. Then we can also develop genetically modified crops that have been used as an alternative resource in industries. So even we can develop GM crops, so that have been used as an alternative, that have been used as an alternative resource, been used as an alternative resource in the industries, in the in the form of starch, in the form of starch, used in the form of starch, and even for fuels, okay, and even pharmaceuticals, etc. So this is what all the significance of genetically modified crops. Okay, so now let's discuss this one, especially how we are going to construct these pest resistant crops by using biotechnology. So let's see first pest resistant crops. Under this, we will study especially the BT crops. So let's see. So while scientists doing research, they have come across a bacterium named Bacillus thuringiensis. Okay. So this bacterium produces, it produces a toxin. So a toxin, which means a poisonous substance that can kill insects. And this toxin is produced by this Bacillus thuringiensis in a particular phase of its growth. Okay, so this toxin that is produced by this bacterium. So this is a bacterium. Okay, and that toxin produced by this bacterium is simply called the Bt toxin. So in this case, the Bt stands for the genus and species of the bacterium from which that toxin is obtained. Okay, and this Bt toxin is produced by the bacterium during its or during a particular phase of its growth. Okay, and the point is that even though it produces this Bt toxin, that toxin, it will be not effective on the bacterium. So the reason is that when it is produced, it will be in the inactive state. So when the toxin is produced, it will be in inactive state. It will be in the inactive state. So later it required certain conditions to become active, and we have come to know that this Bt toxin is very good at acting on some insects like these lepidopterans. Okay, so this Bt toxin is effective on insects like lepidopterans. So these include the tobacco budworm, army budworm. Okay, so tobacco budworm, army budworm. So these are caterpillars of certain kind of insects. We know that these moths, a type of moths, okay, butterflies, they lay eggs on leaves, fruits, stems, and okay, so they leave these eggs on those fruit stems because they are source of food for the young one who emerge out from the eggs. Okay, so when these kind of moths, okay, their caterpillars, we say like tobacco budworm and army budworm, so belonging to, okay, lepidopterans group, right? So this bt toxin can kill these insects, and not only on them. So this bt toxin can be effective even on the coleopterans. This includes beetles. So it includes beetles, and even on dietarians, that includes flies and mosquitoes. That includes flies and mosquitoes. So lepidopterans, on coleopterans, and also on dietarians, this bt toxin is very very effective. So now, okay, so the scientists, what they have done is the gene that codes for this bt toxin. Okay, so say suppose some gene that codes for bt toxin. So that gene they isolated from this bacterium Bacillus thuringiensis, and that gene which have been isolated from this bacterium Bacillus thuringiensis, they introduced into a particular crop, and now that crop is able to produce that bt toxin, and that became a self defendant for this kind of pests. And in that case you need not use pests, so hence there is no use of pesticides to kill these kind of insects. The plant has its own ability to kill them, because you have introduced that gene which codes for the bt toxin into the plant, and the plant is directly producing that bt toxin, right? And like this, already we have produced certain bt crops, like example, the bt crops, example, bt cotton. Already we have produced that bt cotton, then bt tomatoes we have produced, and then bt corn also we have developed. Okay, potato, soya bean, bt potato, bt soya bean, etc. There are now so many, even bt brinjal. There are so many varieties of crops that already we have developed, and they are already in the market. Okay, right. So all these the crops are the one in whom the gene that codes for bt toxin has been introduced. Okay, right. Say suppose let's look at some genes of that bt toxin. So the gene of bt toxin is simply called cry gene. So it's called the cry gene. So it is the cry gene that codes for the bt toxin. It codes for bt toxin, right? And there are some varieties of this cry gene, like we say cry 1AB, then cry 1AC, then cry 2AB. So these are some varieties under the cry gene. So when we see this cry 1AB, it controls corn borer. It controls corn borer. So if I'm going to introduce this gene into the corn, so that insect, which we call it corn borer, will not be able to survive. Okay, and then coming to this cry 1AC. So this cry 1AC is going to control cotton bollworms. The cry 1AC will control cotton bollworms, including this cry 2AB. So this one also controls the cotton bollworms. Okay, so these are some varieties of the genes under the cry gene that all code for bt toxin, and they are effective against which I have mentioned. Cry 1AB is very effective on corn borer. Cry 1AC, the toxin that is produced, is effective on cotton bollworms, including this cry 2AB. Right, now let's look at the action of this bt toxin. So how the bt toxin functions. So action of bt toxin. I told you already that when this bt toxin is produced, it is produced in inactive state. So that we call it prototoxin. Right, I'm sorry, protoxin. So that protoxin is the inactive state. Okay, so when it is produced, it will be in inactive state and it's called protoxin. And to become activated, it requires some conditions, right? So this, in the presence of alkaline pH, it can become toxin. So that is the activated state. So it's an activated state. So the protoxin will become, okay, toxin in the presence of alkaline pH. So now when the plant produces this toxin, it will be in the protoxin state. Now when the insects eat the leaf, say suppose it is present in the leaf, it is present in the stems, not all parts of the plant, because the gene is present in all parts, okay, of the plant, and it's going to produce the toxin in the inactive state. And when the insect eats these parts, okay, so the protoxin enters into its midgut. So here in the midgut, the epithelial cells, so the midgut epithelial cells, this right, so here it says this is the midgut, and these are all epithelial cells in midgut. Now when the, okay, protoxin enters into the midgut, here there is alkaline pH. So in the midgut it is alkaline pH, and in this alkaline pH it's going to become the activated, okay, form, that is toxin. Now this toxin goes and binds to these epithelial cells. Okay, so this toxin, okay, it binds to the, you know, the surface of these epithelial cells. And now what happens is it creates holes on these epithelial cells. Okay, so that's what the activated toxin creates, pores on epithelial cells of midgut. on the epithelial cells of the midgut, right? And when there are pores, you can see here the pores have been created, and because of this what happens, there will be endoosmosis taking place, and the cells will swell in size, and finally the cells swell and burst. Okay, the cells are going to swell and burst means that the epithelial cells are going to die, and because of this, okay, the animal will not be able to digest whatever it eats. Okay, it starves and it dies. So this is how, okay, the action of bt toxin takes place, right? So now this is not only the method of creating pest resistant crops, okay, by transferring the bt toxin gene and making the plants pest resistant. Now there is one more method, so it's called, okay, RNAi. So RNA interference is another mechanism that we used to develop pest resistant crops. So that part I'll be discussing in the next session.